We're going to cover the basic toolkit for biotechnology. There's a lot more information in this chapter 10 of your textbook, but we're going to really focus on what's going on in this PowerPoint so that we're not overwhelmed. Okay, so biotechnology is the artificial methods to modify genetic material of living organisms or cells so that we can produce novel compounds or create new functions. And these are typically used to benefit human well-being. Okay, so the basic toolkit um, are these 3 steps. So we're going to have a nucleic acid extraction that can be either DNA or RNA. We'll usually then move into PCR, which is polymerase chain reaction. So if you've had 23andMe sequencing done, or if you had a COVID test during the early days of the pandemic, there's been some PCR associated with you in your life. Okay, what researchers will usually do then after they have their PCR done is they'll check the quality and quantity of the DNA using this tool called gel electrophoresis. So in your virtual lab for this week, we're going to walk through some DNA extractions where we're basically going to take some cells, we're going to split them open, and then we're going to denature all of the proteins and the other things that might be in the way so that we can then isolate that pure DNA. Okay, so check that out. I walk you through all the steps of how to do that. You can follow along. Or if you want to use this YouTube video that I've linked here in the slides, that will be a different kind of DNA extraction that you can actually drink if you're 21 or older. All right, so let's talk about this thing called PCR. What the heck is that anyway? And PCR is a extremely useful and widespread biotechnological tool that was invented by this dude named Kary Mullis. Yep, that's a Nobel Prize winner's memoir. Yep. All right, so we can use it to figure out the genotype of an organism, to do some genetic cloning that we're going to talk about later. We might use it to determine if there's mutations present and how does that affect the function of the organism. We need to do PCR before we do DNA sequencing, and there's so many more applications. So what is PCR? This is a method where researchers can make billions of copies of a single gene, and that's really important. When Mullis invented this, he was working on understanding the genetic markers around sickle cell disease. And so he was looking at the gene for hemoglobin, that is the protein that carries oxygen and carbon dioxide around in our blood. And people with sickle cell anemia, their hemoglobin doesn't function properly. So he was looking for this one single gene, but all the rest of the genome, all those other genes were getting in his way. And so he invented a method to make copies of only the gene of interest and to ignore all the other parts of the genome. And so that way we can focus the research on a single gene. One cycle of PCR involves these 3 steps. First of all, what we need to do is we need to take that DNA, that double helix DNA molecule, and we need to melt it into 2 single strands. Okay, that's the first step. The second step then is we need to take that primer, this DNA primer that tells polymerase, this is the starting point. This is where you need to make a copy. We need to stick that on to the complementary strand, right? So A's and T's, C's and G's are going to hook together. Okay. And then the last step is where our polymerase is actually going to be incorporating new nucleotides and building that new strand of DNA molecule. We call that extension. Okay. And so then we're going to, so if we started with 1 copy, at the end of the first cycle, we're going to have 2 copies of that gene. And we'll do it again, and then we get 4 copies, and then 8, and then 16, and then 32, and so on and so on and so on and so on until we have billions of these copies of this gene of interest. So after we get our PCR product, we want to check the quality and quantity of that DNA. So what are we going to do? How are we going to do this? We're going to use this tool called gel electrophoresis.